Good morning, our listener. Welcome on our video today. Today we have biology practical. This one is paper 3 KCSE. Today we are in laboratory and we are doing practical. Assume that we are in a practical room, and you can see that we have question number 1. It has 10 marks. Question number 2, 17 marks, and question number 3, it has 13 marks. Total is 40 marks, and we have answers here for you. These are the questions. You can see them. Question number 1. You can see question number 1. This is question number 2. You can see question number 2. This is how they come in KCSE. If you have never seen a paper, biology practical paper, this is how it appear. Today we are doing it one-on-one. This is question number. This is question number 2. Then here, this is question number 3. So we have said we have question number 1, 2, and 3. That is 2. And this one is question number 3. You can see it very well in your screen. So we shall perform this practical, and we are going to get everything. So we request you to watch up to the end so that you can be able to understand how the biology practical paper is done. And if you get maybe out of 40 you get 38, it means that in biology you are going to make it. I hear them say if you pass in practical, then you have passed in that paper. If this is your first time to watch our videos, kindly consider subscribing to this channel. Take 2 seconds and subscribe to this channel. Also, like this video. People are watching biology exams or in YouTube. This video will be recommended to them, and they will also benefit as you are benefiting. Also, we request you to like, share, and subscribe. To the subscribers, we really want to thank you guys. Your support has made this channel to grow the way you can see down here. You can see we are very many, and we continue to grow each and every day. Let's go to question number 1. You are told you are provided with a whisking tubing, a solution labeled K, iodine solution labeled solution H, Benedict solution labeled solution J, and a piece of thread, and you are told A, using the reagent provided, put 2 ml of solution K in a test tube. In each case, test for the food substances present. So what is important here is for you to understand the solutions, because they are not telling you which solution is there. They have just labeled them. They have labeled them K, J, and H. So what is important for you is to be very careful. You should also have some knowledge of what you are testing. Once you see iodine solution, what are they testing? Once you see Benedict solution, what are they likely going to see? Going to test? Which food are they going to test in that specimen provided? So this is how the paper appear. You are given a table like this one. You are told food substance. They are going to test what? Starch. Then you write the procedure. So there is marks for writing procedure, there is also marks for writing observation, and there is marks for writing conclusion. there is marks for testing reducing sugars, marks for the procedure you follow, marks for observation you made, and marks for conclusion. So you should be very careful when testing. You are told tie one end of the whisking tubing tightly using the thread provided with the help of a dropper. Put 10 ml of the solution labeled K in the whisking tubing. Tie the other end of the whisking tubing tightly, and you are to ensure that there is no leakage in both end of whisking tubing. Next, you are told wash the outside of the whisking tubing with water. Place the whisking tubing upright in a 100 ml beaker. Add distilled water into the beaker to reach the level of the liquid in the whisking tubing. Allow the setup to stand for 30 minutes. You are told using 2 ml in a test tube. In each case, test for the food substance in liquid outside the whisking tubing. So the first question number 1, where we are testing the starch, let us look how you are supposed to write the procedure. The procedure for testing starch is like this, as you can see here. And a few drops of iodine solution to solution K. Then what will happen is that it turns blue, black, or it turns blue, black, or blue, black. If it turns like that, then you know that there is what? There is a presence of starch in that food. When you are testing the reducing sugar, you add equal amount of Benedict solution to solution K in a test tube and heat. You can see the procedure, very important. Then if it turns orange, or yellow brown, then you realize that there is what? Reducing sugars present. We were dealing with question 2, whereby we followed the procedure as we stated. Now when you do like that, you are told to write the procedure. Food substance you are testing in question 2 is starch, and also reducing sugar. How do you know whether they are present? So what to do? So you are told using 2 ml in a test tube. In each case, test the food substance in a liquid outside the whisking tubing. So for starch, you are told the procedure is add a few drops of iodine solution to the distilled water from the beaker in a test tube. Then observation are, it retains the brown color of iodine or turns brown. That one shows that there is absence of starch. When you test the reducing sugars, the procedure is add equal amount of Benedict solution to distilled water from the beaker in a test tube and heat. If it turns orange, then reducing sugars are present. And you are told to note the procedure. You are told to mark, the examiner is told to mark only once in a table. If it is wrong, award at B if correct. So you can see the technicalities used during marking. Award the observation and conclusion for reducing sugars to I want observation and conclusion for reducing sugars. Test at A only. If wrong at A, and I want at B, then you are told in C, account for your result in A and B above. A and B above. Why is it like that? A and B. Why is it like that? The visiting tubing is semi-permeable, selectively permeable, hence allows the small molecules of reducing sugars to diffuse, pass through, but not the large molecules of starch tied to A and B. Let's go to the next question. Question number 2. We are through with the question number 1. Number 3, the photographs labeled D, photos, photo D and E. You're told with a reason, state the class of plant from which the leaf in photograph E was obtained. E, where was it obtained? Then you state region, class and region. Let's get the answer. So E was obtained from monocotyledonine. Reason, parallel venation or presence of leaf sheath. Next, you are told state 2 features in the leaf shown in a photograph D that adapt it to its function. How does the leaf, how does the D, how is it adapted to its function? It has broad flat leaf, broad and flat, to offer large surface area for photosynthesis. Number 2, thin to reduce a distance over which carbon dioxide diffuse to reach the mesophyll or photosynthetic cell. Next, rich supply of veins to supply water to the photosynthetic cell. Next, presence of chlorophyll to absorb for photosynthesis. Next, standard paragraph K, L, N and P and Q provided below and answer the questions that follow. Let's see whether we can see the specimen they are talking about. Yes, they are here. Standard photograph K, L and Q. K, L and Q. K, L, Q. K, K1, L1, and yeah, they are here. You can see them. You are told to identify part labeled, identify the part of the photograph labeled K1 and L1. K1 and L1. K1 is a leaf tendrils. L1 is stem tendrils. Next question, you are told state the role played by the part K1 and L1 in the plant. K1 and L1. What role do they play? What role do they play? They provide mechanical support to the plant. Next, you are told to, that is Roman 3, name type of response used by structures K1 and L1 to play the role you have mentioned in Roman 2 above. Name the type of response. What type of response they use? They use digmotrophism. Digmotrophism. It is also called aptotrophism. Aptotrophism. Number 4. This is practical. We are doing practical. Name type. We have seen that one. We have answered that one. Name the hormone involved in the response you have mentioned. Which hormone is involved in that? That one is auxilin. Auxin hormone. C. Go to C. You are told name the type of evolution demonstrated by Name the type of evolution demonstrated by photo K and Q. K and Q. That one is divergent evolution. We go to the next one. What type of structures are represented by your answer in C Roman 1 above? That one is homologous structure. We go to next one. Homologous structure. Go to the next one. Give reason for your answer in C2 above. What reason do we have for that? Both are leaves modified to serve, or both are leaves modified to serve different functions. K is modified to for support or for photosynthesis, while Q is modified for food storage. We are through with this video, and we thank you so much for watching up to the end, and we believe that if they bring a paper like this one, or they bring a question like this one, you are going to get everything out of this. If you have not yet subscribed to our channel, kindly take this moment, subscribe to our channel and like this video. To the subscribers, we want to thank you guys for the support that you've given us. You can see this channel is growing because of the support that you have given us. Today we request one thing, like this video, and this video will be recommended to other lovers of biology. Until next time.